There you go. That is the creature. That is the thing that is the new species of the week this week. It is really heckin' cool. It is really, really heckin' cool. All right, y'all. So what is the prediction for this? What is the result? Folks, interesting. 2% on amphipod, which is one of those aquatic creatures that we see sometimes on Microscopy Monday. None on copepod. 10 points on a plant. Ooh, those plant people. Y'all are, nice. 10 points on a squid. No points on a fish. 22% of the vote on a mushroom. 18% of the vote on a bacterium. 40% of the vote on a hopper, which is a bug. 18% of the vote on a worm. And 10 points, almost 0% of the vote on a frog. Folks, the correct answer is, and it's a brand new species of mushroom in Switzerland. And we'll take a look at the picture. So there is the beautiful brand new mushroom. So it is this Mycenia crocata is the brand new species of mushroom. Potato crotata Croatia. There's lots of these species that have been reported. And chat, you know how we have a lot of people who are, when we talk about the new species of the week, they're a little bit sus, right? They're like, is it really though a new species? Like, is it really different enough to be a new species? How doth one know that it's a new species? Well, Corn Relish, that's a great question. I'm glad you've asked. This one is so fundamentally different that this is the first species of this genus Mycenia that fluoresces. It is the first and only one. And so I feel like we can say it's a new species. If none of the other ones tested have been fluorescing and this one does, like, all right, I'm pretty much okay with this. Yes, yep, plant, animal, fungus, yep, different kingdoms. So there's the plant kingdom, there's the animal kingdom, and there's the fungal kingdom. And interestingly enough, fungi and animals are more closely related than fungi and plants. I'm not sure if they're beyond that you're able to eat it more. There is none of that in terms of toxicity discussed in the paper. They did a fair bit of like characterization of coloring and size so that it could be 15 to 15 centimeters. It's very slender. It has a color gradient from orange to red and at the base, and then yellow to cream color at the top. Like, they went pretty over in terms of the colors of it. And yeah, they were like really looking at the bioluminescent side of things. And it was really cool because they mentioned within the paper, they kind of set the stage for how it was identified. So they were like, we were walking through the forest in a neighborhood and we saw some green light through our special camera. And they were like, oh, this is cool. Yeah, so they measured what light intensity was coming from what region of the fungus. They also grew them up and cultured in the lab, and they actually stayed bioluminescent for 164 days. They did check the spores as well, which I thought was interesting, and identified that the spores themselves are not bioluminescent. They figured out at what point during development of these fungi there are like bioluminescence from it. They suggested, although openly said, we don't know if this is true, but maybe there are other members of this genus that are also bioluminescent. And that would make sense, like that it's not just a single one-off mushroom that can do this. So Sparky, here in this figure, that's the root system. And then here you're seeing the root system glow. Here again, within the tree bark, some of the root system, and then this is like the fruiting body. So this is like the intense part, like overhead. So this is the part they would have seen. That's the part that they would have seen for it. So there are both visible above ground points and underground too, which I again think it's interesting that there is enough of that range of that visibility to it. Yeah. They go through the methodology, I think, quite well. There's a lot of methods papers or the method sections of papers that are not very well done, but there's a clear bit of information put out here. They measure the luminescence, and actually, maybe Sparky, this is what you'd be looking for. So they looked at different regions of the mushroom, and with the different regions of the mushroom mapped out how much light emission there was. And so the stripe base of it, which was on top of it, is what had the best amount of bioluminescence for it. It is a chemical reaction, yeah, Jay, that takes place, and that's what provides this luminescence. So we have a video on not this particular one, but a different species that does something similar. It's similar to like a chemical reaction that takes place in fireflies. So it is a chemical reaction where One enzyme acts on a protein by cutting part of it off. The cutting of an element of it off releases light. And it's what's really cool is it's not a heat based reaction. There is no heat release from this. Usually these chemical reactions that produce light, the byproduct would be heat, but you're not producing heat here, which is great because you don't want to cook your fungus. They are absorbing something which is causing them to glow. So clear of what's the trigger for them to start glowing. They do say here with like a regular camera, the bioluminescence is less sensitive than with the actual like tool. This is this luminometer, which is actually detecting the light emission. It's much more sensitive than a camera, but they're still able to see it. Yeah, with some soy, possibly. I don't know about still though eating it. And then this was again cool is like when they looked at like the root system, and that the root system that went into tree bits actually also then started to bioluminesce. So here they're classifying this. I kind of wish there would be like additional images to it too. Aha! Jay, see, here's the thing, Jay Spites. And then you ask incredible questions, very insightful questions, and the questions that one should be asking. I'm not gonna have it, Jay. I'm not gonna have it. No, but in all seriousness, like that is the moneymaker of a question: is why on earth does a mushroom need to fluoresce? And there has been, Jay, one experiment that has been done to test this hypothesis. It's not great because I can think of a bunch of things that are wrong with it and it needs to be done better. But it's the same question whenever there's a glowing mushroom, right? Whenever you have a mushroom glowing, you could ask that exact same question of why on earth is this glowing? What is the benefit to evolving and spending energy on anything being able to glow? So keep an eye here. It's kind of a fast movement. You can see visibly that there's some glowing in these mushrooms. This is a different species. These are not in Europe. These are in South America. Are there mushrooms with bioluminescence of different colors? Does that make a difference? Would it help us understand why they glow? I'm sure there are. I'm not available, I don't understand or know offhand what other wavelengths of light are emitted. I think there's some with red wavelength that is not visible to us without very specialized equipment. There's also a lot of bioluminescence that involves fungi, but there's some kind of like secondary creature there, like a bacteria, or it's a fungus living in the gut of another creature, and those are a little bit more dodgy because that's probably for the other creature that's more beneficial. So I'm not sure offhand if that is something that's readily known and like how well. If there were different colors in the same area, maybe those would be signaling different creatures. And so there's actually an experiment done a couple of years ago where they did just this. So this is a different species of bioluminescent fungus, and this species like also glows, and they attempted to answer like what exactly, like why they glow. And so they made fake mushrooms, ones that glowed and ones that didn't, and they just wanted to see, okay, do these attract bugs? And if they attract bugs, maybe it's a pollinating factor. The researchers are like, we don't know why it happens. And Jay's like, well, why does this happen? So that's why I say Jay needs to be on like advisory panels. I can already see some of our friendos, like the brows are twitching. I can feel it, right? See in nature, right? So in terms of a matchup, it's closer to what you can get for a matchup than, I don't know, using anything else. But there are some downsides to this kind of experiment, right? Of what you're going to be potentially attracting because of glue odor is going to have an effect. And no, they've not yet figured out the mechanism for how these things work, but it is like it proves much harder to identify than what you're just seeing, like one that they made to suggest at the beginning of the video, or at the end of the video. Build a live mushroom and see if they attract anything, then see if the interested insects are attracted to that wavelength. So I think the issue here, Neil, is that this is an ecology experiment that they were running. And a lot of the ecology experiments are run in the field because the argument is if you run it in the field, then it's going to mimic most of what you would see in nature. If you have cameras on something like this in the field, there's a bunch of other variables that are going to take place, right? So like the position of the mushroom, other plant material around the mushroom, like there's a lot of external variables, and filming them, I think it's difficult to get the film quality, especially that video was 6 years ago. to be able to get and see what exactly is there without disrupting the bioluminescence of the mushroom. And that is a very, very big step. They not only were able to grow them in the lab, but they were able to get them to bioluminesce in the lab, which is apparently not as straightforward to do as one might imagine. If you can get them to bioluminesce in the lab, then everything is a much, much cleaner experiment, right? Because then you're able to demonstrate that, yes, they're glowing in the lab. You can control where they're glowing. You can have them positioned like having here is a glowing mushroom, here is a non-glowing mushroom, side like two like a Y maze test that you can run. There, your difficulty is just identifying what insects would be the potential targets to test. And at least if you do the glue test, you can collect a bunch of different insects and maybe get a range of what's important and what's not important. It can be complicated to do these experiments, and I think both ways there's not an easy win, but there's definitely interesting questions to be asked. Does that make sense, Neil? Like there's just shortcomings in both ways of doing it more on the ecological side of things and then doing it in the more controlled side of things in the lab. I think, again, the best one, start with the ecological one, get your candidate list on what insects might be interested, and then take it to the lab and do it much more controlled. And whether that candidate list be through the glue or trying to do with the camera, maybe modern cameras would be able to work better than the older ones too. Maybe that would be a little bit easier. Introduce the target insects, yeah. But I guess the hard part of that, Jay, that is assuming that you're able to take the insects from the wild and grow them in the lab. So the best experiments would be is if you took the insects from the wild that were attracted, if you could grow them up in the lab, and as you're growing them up, you're able to age match them and age control them. That you then start testing, okay, these are the insects that I'm putting into this container to test, and they're all the same age. Because that's another experiment that's not visible and doable in the field, right? Like you have no idea how old the animals are that are coming to the mushroom. And right, is it something that's on its way out and dying and going to the mushroom, or is it something that's actually a young insect and they're actively going to this site for like an intentional reason? Like those can be difficult questions to answer, and those could be difficult things to try to account for, unless you have them age matched, if that makes sense. And that's again, that's our first science news story, y'all. I thought it was a neat new species. We usually don't get new fungi, but it was really cool to be able to pull out a fungus news story to kick off the stream with. And again, it being like, I don't know, it was glowing. It was neat. So that's the first science news story, y'all.